That sounds great. Welcome everyone to the webinar today, Considerations for the New 4-Month Rifapentine-Moxifloxacin Regimen for Drug-Susceptible TB in the US. I'll turn it over to Olivia DuPont from the Global TB Institute at Rutgers to go through some housekeeping with us.
Thanks, Neela. Yeah, we just have a couple housekeeping slides and reminders for all of you before we get started. So continuing education credits are available for this webinar. There are CME and CNE available. Please note that you will need to complete an evaluation to receive those credits. So we'll be sending out that evaluation by email at the end of the webinar and in the chat box as well. So please just keep your eyes out for those messages. And if you have any questions during the webinar, please feel free to use that Q&A box that's at the bottom of your screen. And if you have more general comments, you can enter those into the comments box, or the chat box, excuse me, also at the bottom of your screen. And the Q&A will begin with pre-submitted questions that were collected during the registration process. But we will try to address any new questions that come up with the remaining time. Okay, Anila, I'll pass it back over to you.
Thanks so much. We have some exciting talks lined up for you today about an incredible study looking at an important 4-month regimen in the treatment of TB disease. We'll be hearing directly from study investigators, starting with Dr. Susan Dorman, who'll be going over study objectives and methods. Dr. Dorman's an infectious disease specialist and professor of medicine at the Medical University of South Carolina, where her research focuses on TB. She previously served on the faculty of Johns Hopkins School of Medicine for 16 years. She'll be followed by Dr. Payam Naheed, professor in the Division of Pulmonary and Critical Care Medicine at the University of California, San Francisco School of Medicine. Dr. Naheed serves as director of the UCSF Center for TB and director of clinical trials operations in the UCSF Office of Research. We'll then hear from Patrick Phillips, assistant professor in the Division of Pulmonary and Critical Care at UCSF and faculty in the Department of Epidemiology and Biostatistics. Next up will be Dr. Richard Chaisson, professor of medicine at the Johns Hopkins School of Medicine and director of the Johns Hopkins Center for TB Research. Dr. Chaisson is also the director and principal investigator of the Johns Hopkins Center for AIDS Research. Finally, we'll hear from Dr. Ekaterina Korbatova, medical officer in the clinical research branch at CDC's Division of TB Elimination. Next slide. In addition to that star-studded lineup, we'll be engaging a discussion with principal investigators from our TB Centers of Excellence, including Dr. Lisa Chen, Medical Director of the Curry International TB Center in Berkeley, California, Dr. Dave Ashkin, Medical Director of the Southeastern National TB Center in Gainesville, Florida, Dr. Alfred Lardizabal, Executive Director of the Global TB Institute in Newark, New Jersey, and Dr. Lisa Armitage, the Assistant Medical Director of the Heartland National TB Center in San Antonio, Texas. And with that, I'll hand the mic to Dr. Susan Dorman.
Good afternoon, good morning, everyone, and thanks so very much for joining us today. I'll start off by providing just a brief summary of the study objectives and methods. Next slide, please. So reducing the length of time for treating pulmonary TB is an important public health goal. And the study that we're looking at today addresses specifically the questions of whether high-dose daily rifapentine, with or without moxifloxacin, allow treatment shortening to 4 months for drug-susceptible pulmonary TB. Next slide, please. I've listed out the pre-specified protocol objectives here to give you a sense of just the entirety of this study. There were primary or main objectives, as well as a number of secondary objectives. The primary objectives listed on the top had to do with efficacy, and they were centered on evaluating the efficacy of 2 regimens, which I'll go over in just a second, to determine whether the substitutions, or in one case, substitution plus addition of moxifloxacin, made it possible to reduce to 4 months or 17 weeks the treatment of drug-susceptible pulmonary TB. In terms of secondary objectives, There were objectives to evaluate the safety, in other words, the adverse events, as well as the tolerability. In other words, can an individual continue to take their medication? In addition, there were secondary objectives that got at other issues, for example, PK and PD, along with other issues to really understand better what the regimen was doing. Next slide. So this slide shows in a pictorial form the regimens that were studied in study 31, A549. Up on the top is the control regimen, which was 6 months long or 26 weeks. That's the regimen that you all know very well. The first investigational regimen that's shown in the middle there, and we've sometimes called the Rifapentine regimen, was a 4-month regimen or 17 weeks. And that had a 1-for-1 swap of Rifapentine for Rifampin. The other investigational regimen is shown on the bottom. That was also 4 months in duration, 17 weeks. That had the same Rifapentine for Rifampin swap, but in addition, substituted Moxifloxacin for ethambutol during intensive phase, and then continued that Moxifloxacin through the continuation phase. Participants were randomized 1:1:1 to these arms. The primary efficacy endpoint was the outcome at 12 months after randomization, but we also followed participants for 18 months after randomization. Next slide, please. Just some additional notes on the study design. Looking at the bottom left, safety labs, which mostly were CBC and differential and creatinine liver chemistries, were done relatively frequently during this phase 3 trial. Sputum was obtained also relatively frequently during treatment as well as after completion of treatment. This was an open label trial for reasons that we can discuss afterwards if you're interested. It was a non-inferiority design. I won't say more about that right now because Patrick Phillips will provide a lot of detail on that upcoming. All treatment was administered on a daily basis, that is 7 days per week, and with direct administration, directly observed therapy for 5 of those 7 doses per week. The Rifapentine was at a dose of 1,200 milligrams once daily for all participants, regardless of weight, and the Moxifloxacin dose was 400 milligrams daily. Rifapentine and Rifampin are different in terms of their absorption with respect to food, and so Rifapentine was given with food, Rifampin without food, in order to optimize the pharmacokinetics of each of those drugs and therefore each of the regimens. Next slide, please. This just is a map of the 34 clinical research sites across 4 continents. You can see that the United States, South America, East Africa, and South Africa are well represented along with Asia. Next slide, please. Next slide, please. Thanks so much. Listed here are all of the eligibility criteria for the study. I listed them all here so that you can see them and really examine them. Let me summarize some of the high points. So we included adolescents and adults greater or equal to 12 years of age and older. They were required to have a positive sputum smear or a positive sputum gene expert with a medium or high result. We included HIV positive as well as HIV negative individuals. Everyone had to be HIV tested. If an individual was HIV positive, then to enter the study, they were required to have a CD4 count greater than or equal to 100. There were a set of chemistry and CBC laboratory parameters that are shown here, and I think on balance were pretty liberal. Women could not be pregnant at the time that they enrolled into the study. And we enrolled individuals with what's called a Karnofsky score of greater than or equal to 60. So these are individuals who met or exceeded an ability scale of requires occasional assistance, but able to care for most of their own personal needs. So who did we exclude from the study? We excluded individuals who'd had meaningful number of days of TB treatment prior to study entry. We excluded individuals who had forms of TB, central nervous system, bones, joints, miliary, pericardial, that we generally would agree require more than 6 months duration of therapy. We excluded individuals with weight less than 40 kilograms, unable to take oral medicines. We excluded individuals with a known history of prolonged QT syndrome. Of note, we did not do EKGs. We excluded individuals with current or planned use of a number of medications that are listed there because of drug-drug interactions, largely with the rifamycins. We also excluded individuals if their TB isolate was already known to be resistant to isoniazid, rifampin, PZA, ethambutol, and or fluoroquinolones. There were also some what we called late exclusions. These are individuals who enrolled in the study, but then were excluded from study participation afterwards for a couple of different reasons. One was if none of their sputums grew Mycobacterium tuberculosis, or if they were found to be resistant to isoniazid, rifampin, or fluoroquinolones. Next slide, please. So the primary outcome of the study was TB disease-free survival at 12 months after their study treatment assignment. So we assigned study outcome status based on a set of pre-specified criteria, such that every participant who enrolled was assigned an outcome. And it was one of 3 outcomes: cure, absence of cure, or not accessible. In order to be classified as cure, an individual had to have a sputum cultures that were negative at the end of follow-up, or they were unable to produce sputum at the end of follow-up, and they were not already previously classified as unfavorable. You might wonder, what is the non-accessible bunch? So not accessible is a set of exclusions that were unlikely to introduce selection bias into the study. For example, women who became pregnant while on study and therefore were removed from the study per the study protocol were classified as non-accessible. There were 2 primary analysis populations that were pre-specified. The first one was what we call the microbiologically eligible analysis population. And sort of in normal words, this is the large group of study participants whose microbiology showed that they were not resistant. In the microbiologically eligible analysis population, individuals who were classified as not accessible were classified as unfavorable. The accessible analysis population was a subset of the microbiologically eligible analysis population. The accessible population excluded that yellow box that says not accessible. Next slide, please. And this is an important concept, and I think both Patrick and Payam will drill down a little bit more on this, but I wanted to introduce the concept early. So our choice of the microbiologically eligible and the accessible populations as co-primary was in keeping with regulatory guidance. And as Payam will describe, we also performed a number of secondary analyses, and those are down towards the lower right, in other analysis populations. For example, in gold at the lower right, the subset of participants who took 95% or more of their study doses. We also did a number of pre-specified sensitivity analyses. And so the idea here is that each of these analysis populations incorporates and includes some bias, but the domains of bias may be slightly different for each of these analysis populations. And the reason to look at different analysis populations like this is to check for, ideally, consistency of results across those populations. If the results are consistent between all of the analysis populations and the sensitivity analyses, then we've got confidence that the risk of bias was low and that the study results are valid. We'll talk through this a bit more later. Next slide, please. And this just shows in sort of a tabular form how we derived each one of these analysis populations. And this is a slide that really merits, sitting down and studying it, which we don't have time here, but I've added it to help you understand sort of the systematic approach that we followed. So next slide, please. Great, and thanks so much. And I think over to Payam now.
Thanks so much, Dr. Dorman, for such a clear presentation. Go ahead, Dr. Naid.
Okay, thanks very much, Susan. So I'm going to, in this next 10 or 12 minutes, just review the results of study 31A5349, which has been published several months ago in the New England Journal. Next slide. So first, insofar as the enrollment itself, just a graphical presentation of the geographic diversity of sites that were involved and enrolled into this trial from between January 25th, 2016 and October 30th, 2018. 2,516 patients were enrolled across these sites. Next slide. Here I'm showing you the consort diagram. This is also from the New England Journal publication, where you can see that about 5,368 patients were screened, and about a 50% enrollment to screen failure, if you will, randomization, which is represents a good performance for trial inclusion and exclusion criteria. And then you can see the 2,516 patients were then randomized to the control arm, the Rifapentine arm, and the Rifapentine-Moxie arm. And what I'm showing you here is the intention to treat analysis population, which is just according to randomization, as shown in the figure that Susan just went over. The next population down is the microbiologically eligible group, and the microbiologically eligible group includes everybody except for those who had evidence of baseline drug resistance or did not have baseline positive cultures for TB, or were enrolled in a violation of some eligibility criteria. So an inclusive population, I would say, broadly. Then additional set of exclusions that Susan also mentioned then lead us to the accessible population shown on the bottom box. And the distinction here is that those who are not accessible, as shown in this white box here, in the microbiologically eligible group would have been characterized as unfavorable, whereas they're excluded from the accessible analysis. And an important point is that to remind you that in order to claim non-inferiority, we had to prove non-inferiority in both these co-primary populations, microbiologically eligible and the accessible group. Next slide. So this is a high-level overview of the characteristics of the patients enrolled in the microbiologically eligible population. 70% on average male sex. This is consistent with other clinical trials as well as global epidemiology in TB. Median age is around 30, 31. There's a diversity of races shown here. We had about an HIV co-infected enrollment rate of 8.2%, which it matches the global epidemiology of people living with HIV and TB. And then cavitary disease was noted in 72% of the enrolled participants. That in conjunction with BMI median of about 19 and the weight shown here suggests that this was a population with moderate to severe degrees of disease. And so the regimen was being tested in a population that had cavitation as a key factor in TB presentation. Next slide. So jumping straight into the efficacy results, I'm going to draw your attention first to the upper panel. This dashed line is the margin of non-inferiority, which is 6.6%. Patrick will review further in the next talk. And this non-inferiority margin was defined in consultation with the FDA and is consistent with contemporary TB trials conducted in the last 15 or 20 years of substitution trials in drug-susceptible TB. And what we're showing you here is that in the two co-primary analysis populations, the accessible and the microbiologically eligible, that the upper bound of the 95% confidence interval of the difference between the two regimens, the control and the experimental regimen, is well below the 6.6 margin. And this is consistent in both co-primary analysis populations. It's consistent irrespective of adjustment for HIV and presence of cavities. This therefore allows us to claim non-inferiority as met for the Rifapentine-Moxifloxacin arm. Conversely, the Rifapentine arm did not meet non-inferiority criteria, and you can see the upper bound of the 95% confidence interval crossing that dashed threshold of 6.6% or being right on top of it. And this is consistent throughout the accessible and microbiologically eligible population. So one regimen met non-inferiority as specified in the analysis plan and the other didn't. Next slide. So now I'm going to go through the three kind of categories that Susan described and give a little bit more detail to them. The first category is a participant was categorized as being favorable or cured, durably cured. And what you can see here is that the total favorable population is about 88% of the total analysis population. And the definition of total favorable is really defined by being culture negative at 12 months. And you can see the performances of the three arms. The control arm has about an 88.6%. The Rifapentine-Moxifloxacin is 86.8%. And the Rifapentine only is 84.6%. There's a minority, very small number of patients who are seen at 12 months, but were not able to produce sputum or had a culture contaminated or unavaluable. And I think that is a testament to the quality of the trial itself, that out of 2,000 plus patients, there's only 34 for whom we were unable to get a reportable culture result at 12 months. It really illustrates the quality. Next slide. So here is the second category of unfavorable. This is the absence of cure. You can see that there's a total unfavorable of the whole population is about 11.9%. And the respective total unfavorable proportions are shown for the arms, control Rifapentine and Rifapentine-Moxifloxacin. And you can see this pattern of essentially 9.6% for control, 2 percentage points higher for the Rifapentine-Moxifloxacin, and the Rifapentine only arm having not met non-inferiority, also being sort of third place. This slide illustrates a couple of things. One is that almost half of the total unfavorables are not TB related. So those are shown in the bottom box there, total unfavorable, not TB related, describing a variety of other components that are from a regulatory perspective and from the conduct of a clinical trial and analysis approach that minimizes bias are sort of factored into the decision to the definition of unfavorable. And the section above is the total unfavorable that are TB related. And the culture positive at or after week 17 is shown there as well as the principal driver of the unfavorable outcomes. Next slide. And then the final category is that which is not accessible, and this is about 109 total participants out of the 2,500 randomized, and the majority of those patients, the not accessible group, are participants who were not seen at month 12, but their last available cultures were negative for TB. In the microbiologically eligible population, this group of participants would be deemed as being unfavorable, to be conservative on that spectrum that Susan showed you. And in the accessible population, this group was excluded from that analysis. Next slide. Importantly, one of the features of this clinical trial, which is, I think, distinct from other contemporary TB trials conducted in the last two decades, is really the overall consistency of the results, no matter how we look at it, however we slice and dice it. And what I'm showing you here is 14 pre-specified, in other words, they were defined prior to the completion of the trial, sensitivity analyses that illustrate that consistency I was referring to. Across all 14 sensitivity analyses, which include various definitions and parameters for culture negativity and cure, and you can see this consistent non-inferiority of the Rifapentine-Moxifloxacin regimen, and conversely, the consistency of the signal persists for the Rifapentine-only substitute arm, which did not meet non-inferiority throughout. Next slide. I'm not going to go over this slide again, only because Susan did really a nice review of this, but just to show in the circle, the red circle, that we have two co-primary analysis populations that were consistently showing the results that I just showed you, and that was the primary endpoint. Next slide. These are the same plots, actually also in the New England Journal paper, now going into sort of beyond the primary co-primary analysis populations that we've been focusing on, the microbiologically inaccessible, but frankly showing you also the intention to treat population, the TB, per protocol 75%, per protocol 95%, which basically means 95% of the doses and intervention was taken according to the way the protocol had required. And even a post-hoc analysis, importantly, this post-hoc means that we didn't pre-specify this analysis, but we did it anyway upon request from the New England Journal. And you can see again the consistency and robustness of the results of the Rifapentine Moxie arm. Conversely, you can see the Rifapentine arm did not meet non-inferiority throughout. Next slide. One of the questions that we get asked is, where does this place this regimen in the context of other 4-month regimens that have been tested in recent clinical trials? And this is thanks to Patrick Phillips for developing the slide. What he's done here is showing the microbiologically eligible analysis populations of the several recent phase 3 clinical trials, registration quality trials. And it shows that the Rifapentine-Moxifloxacin regimen is well, it's the only one that's met non-inferiority, so it's at the top. It is the most potent we've ever studied in history as a 4-month regimen. The Rifapentine regimen, whereas it didn't meet non-inferiority, as you can see here, it still was the second most potent regimen studied to date as a 4-month regimen. And the comparators here are the Fluoroquinolone substitution regimen studied in Remox and Rifaquin, and then the predominant Moxie pyrazinamide regimen studied in STAND. So, I think this is sort of has opened an opportunity, if you will, to now achieve treatment shortening even beyond 4 months. This is like the first part of the journey to continue our march down to shorter, safer regimens. Next slide. So with these data, the WHO convened a guideline development group in June of 2021. They released a rapid communication and stated that a review of the evidence by WHO has shown similar performance of a shorter treatment regimen compared to the standard current standard regimen, both in terms of efficacy and safety. And they note some of the advantages of the 4-month regimen and have endorsed this regimen as one that can be used worldwide. Next slide. They do note in the rapid communication that there are some clearly, as would be with any new regimen, some implementation and uptake aspects that need to be sorted out. They raise the issue of cost of rifapentine and availability of rifapentine broadly in the international domain. And then they also raise the need for antibacterial stewardship, which frankly is foundational to infectious disease, as you all know. And they point that they raise the issue around being mindful of the use of a fluoroquinolone in the first-line regimen, particularly as it pertains to its value in drug-resistant TB. Very valid implementation and scale points are raised here. Next slide. So in summary, the 2-month HPZM, 2-month HPM regimen met non-inferiority criteria for efficacy in all primary and secondary analysis populations, all sensitivity analysis. I didn't show you, but they were consistent in all subgroup analyses, including people living with HIV, children, adolescents, and in diabetics. The consistency was there in terms of the regimen performance. WHO Guideline Development Committee has endorsed the regimen, and we are awaiting a sort of a final detailed recommendations later in 2021. That will be in updating the module 4 of the consolidated guidelines from the World Health Organization. Next slide. Just a quick slide on the protocol team involved in the design and execution of the trial. Next slide. And then our thanks to the funders, CDC, NIH, the CDC Data Coordinating Center, Division of TB Elimination. Drug supply and TB PK testing was provided by Sanofi. The DSMB was at TBTC, and we thank the staff across the 34 clinical trial sites on 4 continents, the 2,516 participants, their families and friends, as well as the invaluable support and input from Community Representation Advisory Group and the Treatment Action Group. Thank you.
Payam, what an incredible feat you and your fellow investigators accomplished with this well-designed international study. At this point, in thinking about what this might mean for US TB programs, I'm going to go ahead and bring in Dr. Lisa Chen. Lisa, we're seeing comments here in the Q&A, one from Dr. Nilsson about the high percentage of cavitation, another question about patients with diabetes enrolled. As a direct advisor to so many of our TB practitioners in the field, do you think the study's patients are representative of TB patients that we see in the United States?
That's a great question, Neela. I want to say I think that it is impressive how much disease these folks had, and that the young population, extensive disease, the low BMI, the HIV involvement is great for the global population that's going to be most affected. But I do understand that a lot of folks listening today are from programs who are going to have an older population, a lot of comorbidities, a lot of diabetes, as someone mentioned in the Q&A. So I know that you guys just hold on, because I think that Dick Chasen and Katerina are going to address that data. And I think, we're going to be able to take a lot out of the study, even though it's not exactly who your program treats.
Thanks, Lisa. It looks like the study screened for and excluded patients who are found to be resistant to INH, rifampin, PZA, ethambutol, fluoroquinolone, or who had cultures that failed to grow MTB. Is upfront resistant testing something that you're aware of is currently happening at all, or would be feasible in a US TB program setting?
Right. I know a lot of you are thinking, oh my goodness, I have to get expansive rapid moleculars right away. Everyone has access. Everyone has access to the CDC's MDDR. And then if you're lucky enough to have a regional referral lab that you can send to quickly, like the folks here in California, the low incident states, go to Richmond, and other referral labs around the country. You've got it available, but I know the reality in programs that the mechanism isn't always there for it to happen in a timely way. So I think that if this becomes more and more your norm, those protocols and those kind of access links are going to need to get solidified. So, and the labs have to be ready for a larger influx of asks for more expanded rapid moleculars, more than just your gene expert. So yeah, Neil, they have access, but we're going to have to work at it.
Yeah, we're going to see some evolution. And maybe this is a question for you or study investigators, but some of the pre-registered audience asked, why was Moxifloxacin chosen over Levofloxacin, and could these fluoroquinolones be used interchangeably?
Yeah, I can address that. So there were practical concerns and then also some conceptual issues. From a practical level, the TB Trials Consortium had previously studied Moxifloxacin in its phase 2 trials. So study 31 was a follow-on to that. And additionally, in the mouse trials, Moxifloxacin had been used. So there was no compelling reason for us to change. It's also important to note that there's no compelling evidence that one, either Levo or Moxie, is better than the other. The other conceptual underpinnings for using Moxie come from some of the lesion, especially the animal lesion PK/PD work that's being done that shows that Moxifloxacin penetrates into relevant parts of pulmonary lesions in a way that Levofloxacin appears not to.
So I just want to jump in there because, Susan, I think what was important, and I think what people are going to want to hear is just what you said, that so far there's not data in humans that show one better than the other. So I know a lot of people, you choose Levo or Moxie, some of it based on comorbidities, concerns that you have with side effects. And I think most people would say in this case, from a practical standpoint, when you apply this, you're likely to be able to switch unless, I don't know, Payam or Susan, you feel that there's a reason where they'd have to drop out of this regimen if they had to use Levo instead of Moxie. But I think most people would feel comfortable changing it out if you had a good, solid clinical reason.
I'm not entirely.
Or is that hard to say?
I think our study did not evaluate Levofloxacin. Right. And again, in the animal models, there are clear differences between the penetration of Levofloxacin and Moxifloxacin into what we think are clinically relevant pulmonary spaces. So in my own opinion, in my practice, I would not feel comfortable substituting Levofloxacin. The study used Moxifloxacin, and I think until there's evidence otherwise, Moxifloxacin to me would be the preferred fluoroquinolone.
Well, that's important, I think, for folks to hear from you folks, because I know you guys are clinicians as well. I think, in practice with the MDR cases, I know the animal data with the, increased bactericidal activity as well, with the Moxie, but there haven't really been a lot of head-to-head data. And I think that from a practical standpoint, in terms of outcomes for MDR treatment, we interchange the two. But I understand the caution with this change and with this new regimen. So, but it's an important topic to bring up because from a really practical standpoint, people are going to face this as they roll it out in their programs.
Yeah, I echo that. No, thank you both, Lisa and Susan, for expressing those perspectives. It's really valuable. For the moment, we're going to keep going and hear a bit more from Patrick Phillips about why a non-inferiority approach was used in this study to help us interpret results, and then we'll continue to tackle some more Q&A. Dr. Phillips?
Thank you, Neela. Yeah, so thank you for the opportunity to talk a little bit more about non-inferiority and to try to help you to understand what this means and how we interpret that. Next slide, please. So just to recap on the results that you just heard, the control arm was a 6-month, rifampicin-based regimen. There were 2 4-month regimens. The rifapentine-only regimen, which I'll call just the rifapentine regimen, was not non-inferior to control, and that was consistent across a number of analyses, primary, secondary, sensitivity, as Payan showed. On the other hand, the Rifapentine-Moxifloxacin regimen was non-inferior to control, consistently across a number of analyses. Next slide, please. Now, you'll be familiar with a sort of standard superiority trial where there's just a single comparison. In fact, you may be surprised to hear that in our treatment shortening non-inferiority trial, there were 3 different efficacy comparisons, which I'm going to take you through. And I hope this will help you to understand the context of non-inferiority. So the first comparison was this: is the novel 4-month regimen better than no treatment? This is an indirect comparison. We, of course, didn't have a no treatment arm in the trial. I mean, the external data for this evaluation. Next slide, please. The second comparison, is the novel 4-month regimen better than a 4-month rifampicin regimen? Okay, so this is also an indirect comparison. We didn't have a 4-month rifampicin regimen in the study, and we'll use external data for this evaluation. And the third comparison, is the 4-month regimen, the novel 4-month regimen, at least as good as the 6-month rifampicin regimen? Now, of course, this is a direct comparison. This is what the study evaluated, and we'll look at that one at the end. So next slide, please. So the first comparison, is the novel 4-month regimen better than no treatment? Next slide, please. Now, we need external data to assess this. We can go back to some previous papers. There's a paper there at the bottom. You won't be able to see the full citation because it's been cut off, but in the slides that will be circulated, you can see that. We can estimate untreated tuberculosis about a 50% cure. That's probably optimistic. HRZE, the standard 6-month regimen, about a 90% cure. Okay, so we're looking at a difference there of 40%. Okay, so our question here is the Rifapentine-Moxifloxacin regimen better than no treatment? Next slide. So the difference from 2-HRZE, 4-HR from study 31, A5349, is a 2% difference, and a 95% confidence interval went up to 5.1%. And you may be able to see, I don't think my pointer works, but I've plotted the confidence interval, little black line there up next to the 90%, showing you where this falls. So we have strong evidence. There's not much of a thank you, and not much of a reduction from 90%. So can we say the Rifapentine-Moxifloxacin regimen is better than no treatment? Yes, we can. Next slide, please. Now, is the Rifapentine regimen better than no treatment? Next slide. You can see I plotted the confidence interval on there. It's wider, but we can see very clearly that it's well above the no treatment bar. And so is the Rifapentine regimen better than no treatment? Yes, it is. So we've got evidence from the study to say that. Next slide, please. I recognize that that's perhaps not the comparison you're most interested in. I guess that's a given. Let's look at this second comparison. Next slide, please. Is the novel 4-month regimen better than a 4-month rifampicin regimen? Now, you may be asking, why is this comparison important? And let me just try and take you through the logic here. Now, we know that a 4-month rifampicin regimen is not used anywhere in the world. It's not used in the US. It's not used anywhere else in the world currently. Now, if a novel 4-month regimen is not better than a 4-month rifampicin regimen, it's not going to be used. And so for this reason, we require evidence that a novel 4-month regimen is better than a 4-month rifampicin regimen. Next slide, please. So as before, we need to get external data to answer this question because we didn't have the 4-month rifampicin regimen in the trial. Indeed, it would be unethical to randomize patients to that regimen because we know it's inferior. But there have been previous studies looking at the 4-month rifampicin-based regimens, 2 in particular. This is a sort of summary pooled analysis that's from the REMOX statistical analysis plan. You can follow the link there, which showed that from these 2 trials, historical trials, there was about 11.8% relapse in the 4-month regimens and a 2.9% relapse in the 6-month regimens. This is a difference of 8.9%. Okay, so that's a difference in relapse between 6 months of rifampicin and 4 months of rifampicin, 8.9%. And so this was used to justify a 6% margin of non-inferiority for some of the trials that were published a few years ago, REMOX, Rifaquin, Oflatab. And the same argument was used to justify a 6.6% margin of non-inferiority in study 31, A5349. So sufficient evidence that it's going to be better than a 4-month rifampicin regimen. You can read the full justification if you're interested. It runs to 2.5 pages in the analysis plan, which is available as a supplement to the paper on the New England Journal website. So you can look at it there. Next slide, please. Okay, so if I plot the bars like I did with the previous comparison, okay, so we have the 4-month rifampicin regimen, the left-hand bar, the 6-month rifampicin regimen, the right-hand bar. So it's a difference of 8.9%. That's the black dotted line. And you can see the margin we chose of 6.6% is less than that difference. Okay, so the question again, is the Rifapentine-Moxifloxacin regimen better than a 4-month rifampicin regimen? Next slide, please. So I can plot the bar. You can see that little black line. The upper bound is 5.1%, which is less than the margin. So we can say yes. Next slide, please. And just to restate that clearly, the upper bound of the 95% confidence interval does not exceed the margin of non-inferiority, which is 6%. Therefore, study 31, A5349, does provide evidence that the 4-month Rifapentine-Moxifloxacin regimen has superior efficacy to a 4-month rifampicin regimen. Next slide, please. So, next question, is the Rifapentine regimen better than a 4-month rifampicin regimen? Next slide, please. Now we can plot our confidence interval on there, and you can see that it crosses the margin, our pre-specified margin. The upper bound is 7.7%. So next slide, please. So let me just try and say this in words. I've written it down there. The upper bound of the 95% confidence interval exceeds the margin of non-inferiority, 6.6%. Therefore, study 31, A5349, does not provide evidence that the 4-month Rifapentine regimen has superior efficacy to a 4-month rifampicin regimen. So we don't have evidence from this trial that 4 months of the Rifapentine regimen is better than a 4 months of rifampicin regimen. Really, that's how we interpret non-inferiority in this context. Next slide, please. So we've come to the final comparison here. How does I sort of rephrased at the bottom there, how does the novel 4-month regimen compare to the 6-month rifampicin regimen? And we're focusing here just on the Rifapentine-Moxifloxacin regimen based on the last slide I just showed you. Next slide, please. So now, this is a direct comparison. We don't need to consider external data because this is the comparison in the trial. And we can just look at the results as they are. Next slide, please. And I would say to you, really, that the margin of non-inferiority for this particular comparison is less important. So I've taken the line out. I sort of put a little red box over the margin there. And really, we're just going to look at the results as they are. It's important to look at the pre-specified primary outcome and the pre-specified primary analysis populations here, as Susan and Payam indicated for us, the accessible and the microbiologically eligible. And I recognize this is a mixture of outcomes, but the reason we focus on this is because it's pre-specified, so it's accepted by the FDA, the CDC, investigators, the IRBs, as the outcome that we should use for the primary interpretation. But the other thing is to say that whilst this is a combination of outcomes, the other events, the sort of non-bacteriological events that happened, might have been precursors to bacteriological relapse. And so ignoring them, just sort of throwing them out, would have led to an underestimation of unfavorable outcomes. This is why we use this composite outcome. So next slide, please. The question is, how do we interpret this? And I want to talk about number needed to treat, about how we go about doing this. And I've just got a couple more slides to go through here. How do we think about this comparison between a 4-month Rifapentine-Moxie regimen and the 6-month rifampicin regimen? So the number needed to treat is used across disease areas. It's often presented in publications. Many of you will be familiar with it. It's the average number of patients who needed to be treated to prevent one additional bad outcome. So a minimally effective treatment has a high number needed to treat. And what that means is that more patients need to be treated to prevent one bad outcome. On the other hand, a highly effective treatment has a low number needed to treat. And so this means fewer patients need to be treated to prevent a bad outcome. So if you think about sort of hypothetical perfect treatment for a uniformly fatal disease, as compared to placebo, where everybody would die, this treatment would have a number needed to treat of 1. So that would mean one bad outcome is presented for every single patient treated. Next slide, please. It's maybe easier once we actually look at the results. When we think about number needs to treat for a non-inferiority trial, it's important to understand that the novel regimen is the comparator. That's the best way to think about number needs to treat in this context. And so the way we think about this, for a non-inferiority trial, we consider number needs to treat as follows. So suppose you're considering introducing the Rifapentine-Moxifloxacin regimen. Now, I hope that this isn't too much of a stretch, because I think this is where really we're all thinking about at the moment. Suppose you might consider introducing this novel Rifapentine-Moxifloxacin regimen, okay? So the question is, how high would the number needed to treat of the 6-month Rifapentine regimen need to be to prevent you from doing so? Next slide. Let me show you the results. It may be easier for you to sort of get a handle on this. So for the Rifapentine-Moxifloxacin regimen, as compared to the standard 6-month Rifapentine regimen, the absolute difference was 2%. And this corresponds to a number needed to treat of 50. Now, if you haven't followed anything up till now, just focus on the sentence there in the red box, please, because this is an important sort of summary of this particular comparison. So compared to the 4-month Rifapentine-Moxifloxacin regimen, you would need to treat 50 patients with the 6-month Rifapentine regimen to prevent 1 additional relapse. That's a number needed to treat of 50. Okay, and so this is, I think, it's a helpful way of thinking about this. So this corresponds to an extra 100 months of treatment to prevent 1 relapse. Okay, so 2 months of extra treatment for each of those 50 patients. And so I think these are the sort of questions that we should be thinking about. So what is the cost to the health system of 100 additional months of DOT? What is the cost to the patient of an additional 2 months of treatment? And then what is the cost to the patient, to the health system, to the community of 1 additional relapse? And those are the ways I think we need to weigh up the risks and benefits there. So if we look at the extreme upper bound of the 95% confidence interval, just to think about how bad this could be, compared to the 4-month Rifapentine-Moxifloxacin regimen, it could be as few as 20 patients that would need to be treated with a 6-month Rifapentine regimen to prevent 1 additional relapse. So the lower bound of this number needs to treat is 20 patients based on our results. Next slide, please. So this is just, I'm just restating the conclusions from each of these 3 comparisons. In the interest of time, I won't restate them, but just thinking in terms of those 3 comparisons that we think about in a non-inferiority trial. And that's my final slide. Thank you very much.
Thank you, Dr. Phillips, for such a nice overview of non-inferiority and explanation of how that can help with interpretation of the study results. Dr. Dave Ashkin, I know in your many TB roles, you not only do service on the TB Trials Consortium Data Safety Monitoring Board, but you also do medical consultations for the Florida TB program and provide clinical consultations for the Southeastern National TB Center. Can you speak to your interpretation of these study results from a US TB program perspective with some of the statistical principles that Dr. Phillips outlined?
Yeah, first of all, Neil, thank you very much for the opportunity. And what a great program so far, huh? Pretty cool. You know, I got to say that this is something that's been haunting me. You know, for the longest period of time, all we've been asking for TB is how to shorten the regimens, And we've tried numerous times and it's failed. You know, what's a landmark is this one looks like it was non-inferior. We'll talk about that in a second. Patrick did such a good job. But the bottom line comes down to, you know, when I was sitting on the DSMB and I was looking at these statistics, the whole time, all I kept thinking about is, this is a great study. It is a great study. But how are we going to interpret it? Like, I knew from a statistical standpoint how I was going to interpret it, but I was always thinking about, as a TB medical director, as a TB controller, how do we implement it? And I think that's where we're at this very moment. I think there's 3 different parties we're looking at. We're looking at these results from the perspective of the patient, and from a patient's perspective, 2 less months, that's good. From a clinician's perspective, it's great too, because, you know, we can shorten the regimen. It would be easier to sell to patients. But from the public health perspective, from the public health leader's perspective, that was the one that intrigued me the most. Because I think we were really looking at this point, you know, what does this mean? And from a public health standpoint, the thing that really got me was the idea of the failures, the microbiologic failures. You know, if you look at what Patrick talked about, he was right. They looked at this as being non-inferior from a cumulative background, from looking at people who were lost to follow-up, from people who failed because unfortunately died. But as a TB controller, or we're really set on how well does this work from a microbiological, meaning once they become culture negative, they stay culture negative, a true success. And one of the biggest concerns I have with this regimen has been this failure in microbiological. And just to show you what I'm looking at is that this was the outcomes of unfavorable. And as you can see, if you look at the control group, and I gotta say, Rifa's always perform really well. If you look at only 1% of patients, only 1% of all patients who got right actually failed, had 2 cultures that were positive. Whereas you look here with the Rifapentine-Moxie, 34, 3 times the failure. And those are the kind of questions we have to ask ourselves. The first one being, how much failure are we willing to accept? And when you look at failure, there's a lot that comes with it. It's not just the patient fail. But now you're looking at things like, now you have to restart the patient. You have to work them up. You have to do the diagnostics. You have to retreat them. But also there's other things. Now all the patients you exposed at that point to that person while they were infectious. What about the contact investigations that you have to do? And to me as a TB clinician, the number one thing is the loss of credibility. It's very hard to go back to a patient and say, you know what, it failed. So, I got to say to you, from the big perspective of things, 95% of patients over that, right, will do better no matter what you do. And that's the great news. The great news, the vast majority of patients will do better, will succeed no matter what you choose. But the bottom line is that we have to deal with this. How much failure as TB controllers are we willing to accept? And that's something I think is going to evolve over time.
So Dave, if I'm understanding what you're describing, even though there was non-inferiority seen between the 4 and 6 month regimens observed in this study, there were some differences, albeit small, and you're speaking to the potential impact of these differences, then what do you see as the role of this 4-month regimen in TB programs?
They have to look at non-inferiority very closely. It's different. I keep hearing people say it's as good. It's not as good. It's just not as bad. The whole idea is this regimen, is not as good. As a matter of fact, in statistical terms, we would say this is a non-inferior inferior. We have to understand it's not as good. And the bottom line comes down to is that, we're used to actually non-inferior studies where what we use is actually better. Like 3HP actually was better than non-inferior. But even with all that, there is a role. The role is, I think there's certain populations where I think there is a real use for this, like individuals who you don't have as much time, like either like places like in jail where it's difficult, or places even like in homeless individuals, or people who have to travel, like immigrants and stuff, where this really may be very helpful. At the bottom line, at the end of the day, what this is another tool for us, a powerful tool for us to use in the fight against tuberculosis. I don't think that we, at least personally, at least not right now, would I be recommending this for all of my patients, but I definitely think it has a role. And I think the benefits of this study are obviously very apparent.
Thanks.
And very quickly, for time, we do want to move to Dr. Chasen, but before that, for just a minute, Payam, can you provide some more details about patients in the unfavorable outcome category in the study? I know you went over it in quite detail, but just to kind of circle back to this, if you're willing.
Be happy to, Neela. Just perhaps first, before going into the unfavorable breakdown again from the trial, I'm just in reflecting on David's points, and I'd be interested to hear the discussion in chat and Q&A, and also Dr. Chason's opinions. I think one of the points I was making around the population enrolled in this trial and the sort of level of severity of disease, which I pointed out, one of the indicators was cavitation, was 70% of the population enrolled in this trial. And I don't think I got this message across, was that this regimen met non-inferiority despite a population that had disease that's higher burden or on the higher levels of burden than perhaps some of the US TB control programs see. I mean, we know California's data and New York's data on cavitation is around 20, 22, 25% of the population has cavitary disease. And so I think that tells me that this 4-month regimen is robust, even when faced against severe disease. So this is a real innovation. This is one of the actual biggest celebrations out of this trial, is that treatment shortening is feasible. Up until this trial, as I showed you in comparisons of others, recent trials conducted, we had not broken the 4-month threshold. We finally have. And this will provide us enormous learnings around drug exposure, tolerability, the role of adherence, and the ability to get to shorter than 4 months. So this is kind of a new, the gate has been opened for new innovation and further advance. So from my perspective, I would say that in response to Dave's comments, that these are some of the questions that a guideline committee has to ponder and discuss in terms of additional considerations. But the regimen itself is proving to be robust even with severe forms of disease. Insofar as the unfavorable outcomes in the trial, as Patrick pointed out, they're composite. That's sort of one of the requirements of regulatory perspectives to avoid biases, falsely excluding people who may have had a TB-related outcome, unfavorable outcome, but maybe were removed from some other reason before that event. So I don't know that we'll gain much more than that insight from the unfavorables to address David's points, but the trial just shows that the regimen is robust.
Thanks, Payam. That's great perspective, and we appreciate it. With that, we're going to move to Dr. Chason. Dick, can you talk us through some of the subgroups evaluated in this study?
Sure, thanks. But before I do that, I'll just add a comment on to Payam's response. With a control regimen that works so well, we have very few choices. We can't look for superiority. That's really unlikely to be achieved. And so we're forced to look for non-inferiority. And when you think about non-inferiority, you want to weigh the other benefits of a new regimen, like shorter time to complete treatment. If we look at the standard of care that we currently use that David likes so much, and we compare that to what it replaced a streptomycin-based regimen with an ethambutol-containing regimen. And the difference is about 2 to 3% more relapses with the ethambutol-based regimen. That's the standard of care, but everyone thinks that that's an acceptable difference. So we're talking about very small differences and very big benefits to the shorter regimen. So I think we need to keep that in perspective. So I'm going to talk about the subgroups. So if we could have the next slide, just to reiterate, the inclusion criteria were smear positive or expert positive TB, age 12 and up, so adolescents included, HIV positive if the CD4 count was above 100 with efavirenz-based treatment. We excluded prior treatment, of course, but we excluded some extrapulmonary TB and people with very low weight. Next slide, please. And we had some key subgroups that we included prospectively in our statistical analysis plan to look at specifically: people with HIV, adolescents, people with diabetes, cavitary disease, and then a number of other microbiological considerations as well, and then composite measures of disease burden. So next slide, please. As has been shown before, the population was globally representative of TB patients around the world, a 2-to-1 male to female ratio, which is very typical of TB globally. About 8% HIV positive, very representative of the global situation. 73% with cavitation, so we know that's a little higher than in this country, but as Payam says, it shows that these were patients who really had TB and were really sick with it, and a variety of body weights, and then a small number of people with diabetes that's cut off at the bottom. You can't see where it says that's diabetes on the lower line. So if we could have the next slide. So we did subgroup analyses looking at a number of these features. And so here we're looking at the Rifapentine regimen, no Moxie here, versus control. And there was evidence that there were some interactions. And so what you can see is that for female sex, the Rifapentine regimen worked better than it did in males. But you can see overall no significant differences in any of these other categories. If you go to the next slide, when we look at the burden of disease, we can also see that for people who had no cavitation, for people who had a negative smear, and for people who had a longer time to positive in midget, so lower bacterial burdens, the Rifapentine regimen performed better. So there was an interaction with those characteristics. Next slide, please. For the Moxie regimen, I've shown all of those on this one slide because there is no evidence of an interaction with any of these categories, subgroup categories of HIV, sex, BMI, and then the burden of disease and the outcome. I will highlight the HIV status in a moment, but overall, the Moxie Rifapentine regimen performed extremely well across the board in all of these groups. So next slide. If we look more carefully at the HIV patients, you can see we had about 200 HIV positive individuals. So they were more likely to be female than the HIV negatives. There were larger proportion of females. They were more likely to be Black or African. Very few African Americans, but mostly Black Africans or Black from other regions. They had cavitary disease, and so they were very otherwise similar to the HIV negative patients, but they had HIV. So next slide, please. So what we're looking at here is the success rates at the top of the slide, the efficacy outcomes in the microbiologically eligible and the accessible. And what you can see, interestingly, is that the Rifapentine-Moxie regimen had more successful outcomes, favorable outcomes than the control regimen, and considerably more than the Rifapentine-only regimen. So for people with HIV, the Moxie regimen actually did a little bit better, not significantly better, but it was better. And the Rifapentine-only regimen clearly performed much more poorly than the Moxie or control regimens. Next slide. Now, we also wanted to look at the use of antiretroviral agents. We did this in a staged fashion. So we had two sub-studies, the Efavirenz 1 and the Efavirenz 2 study. The Efavirenz 1 study, at the beginning of the trial, we enrolled people who were on Efavirenz for HIV but developed TB anyway, and they were suppressed on that Efavirenz-based regimen, and we treated them and we evaluated the data. And when we found that the two treatments could be co-administered safely, then we enrolled the Efavirenz 2 regimen, and that was individuals who weren't on antiretroviral therapy but were starting after their TB diagnosis, and we went through the same process. So if we go to the next slide, the Efavirenz 1, these are people who were on Efavirenz and started the experimental regimens, and what we found was that Rifapentine did not affect the concentration, the trough concentrations of Efavirenz during the course of co-administration and had minimal, non-significant effect on clearance of efavirenz. And then if we go to this next slide, this is the group that started their efavirenz after being on TB therapy. And once again, no impact of the Rifapentine regimens on efavirenz concentrations for people starting their antiretroviral therapy. So the conclusion is you can safely use this new novel regimen with high-dose rifapentine and Moxie in individuals on efavirenz-based therapy. Now there is an ACTG trial 5406 that is in development and will be starting probably sometime within the next year, that's looking at the Rifapentine Moxie regimen with dolutegravir. But right now we have no data on dolutegravir. Next slide, please. So what about adolescents? This was a really novel feature of this study, that adolescents 12 years and older were enrolled in the study. There weren't a lot, 67 you can see. But overall, from a safety point of view, there was nothing of concern compared to the control. Very well tolerated. And if we look at efficacy, again, no, no surprises. This regimen appeared to perform as well as the control in adolescents. Next slide. So diabetes, again, small numbers of individuals with diabetes, defined as a diagnosis of diabetes on treatment or diagnosis at enrollment in the study. Once again, safety similar. Actually, you can see there were more AEs in the control regimen than in the experimental regimens, but small numbers. And then efficacy, again, the Rifapentine regimen, these are now showing poor outcomes, unfavorable outcomes, and there were more unfavorable outcomes in the Rifapentine only arm than in the Rifapentine Moxie arm, which was similar to the control arm. So next, just to conclude, our subgroup analyses showed that the 4-month Rifapentine Moxie regimen is non-inferior to the 6-month standard of care in primary and secondary outcome analyses in all of the populations. It seemed to be effective in people with HIV, diabetes, and more extensive disease, as well as adolescents. There were no important drug-drug interactions with efavirenz. The Rifapentine regimen was not non-inferior across the board, but did perform adequately in women and individuals with non-cavitary disease or those with lower disease burden. And there were no differences in safety in any of the subgroups. Next slide. And I think I also, like everyone else, want to acknowledge the team that did all of this work. And then the next slide, once again, to acknowledge the entire study infrastructure that made it possible. Thanks.
Thanks, Dr. Chason, for such a nice overview of findings by subgroup. Dr. Lardizabal, I know that you see patients in your New Jersey clinic and also advise TB docs in your region. If you were to begin an implementation protocol in your own clinics, based on some of the subgroup data that was discussed, would you start all your patients on this regimen, or would you start with a more limited pilot group in a real-life setting, or are there patients you might worry about with this regimen?
Yeah. Well, thank you, Neela. And before I answer the question, I'd really like to express congratulations and gratitude to the study team. Excellent work, both from TBTC and ACTG. An elegant study and something we've been pining for many years. So I personally welcome this study. I think it's an important step forward. As Payam said, it opens the door to shorter course treatments for sensitive TB. So my answer to that question would be yes, without hesitation. I will consider the shorter course regimen. Importantly, the study had enrolled adolescents, an important group of people, which is usually, these patients are usually excluded, and that's great. We can consider down to 12. I would obviously have prerequisites. I'd like to know right away sensitivity to Rifapentine, INH, and Moxifloxacin, and no contraindications, no pregnancies, and female patients willing not to be pregnant during the 4 months. I understand Dave's concerns. There's a glaring difference in the microbiologic failure between at 17 weeks between Rifapentine and Moxie versus RIPE. But again, a step back, and this is a global population who are sicker, it seems. I have more confidence that we'll have better results in our cohort here. Perhaps we can look more closely at who were these failures, or as a corollary, maybe look at how the US group performed and analyze that group. So yes, I think I'll be generally ready to get on with this shorter regimen.
Thanks, Al. And we'll keep marching along. Dr. Gorbatova, we've been getting some questions about safety and the new regimen. Can we hear from you about adverse events seen in the study?
Okay. Good afternoon, everyone. So now we are switching to the full report about safety. Next slide, please. Here is another consort diagram focused on safety analysis population, which included all participants that were randomized in the trial who received at least one dose of the study drugs. Only 10 participants never started study medication and were excluded from the safety population. And as you can see, these excluded participants were equally distributed across the arms. 2,510 participants were included in safety analysis population. Next slide, please. Adverse event reports were collected in all participants, regardless of the study arm, during full duration of the study follow-up. Protocol-specified safety laboratory tests were done at every study visit till study week 22. And as Dr. Dorman already highlighted, at baseline and weeks 2, 4, 8, 12, 17, and 22, every participant had LT, bilirubin, creatinine, hemoglobin, WBC with differential, and platelets done. At baseline, all participants were screened for diabetes, had albumin, potassium test, and had HIV test with CD4 and viral loads in case HIV test was positive. All women of reproductive potential had a pregnancy test, and no ECGs were required. Next slide, please. AEs that required reporting to the sponsor were defined as serious adverse events, or SAEs, all grade 3, 5 AEs, and any new diagnosis, regardless severity grade. Severity grading of AEs was done using common terminology criteria for adverse events, or CTCAE version 4.03. Each adverse event was rigorously reviewed by the CDC safety officer and coded in standardized term using MEDRA. Even though Study 31 was an open label trial, only DSMB reviewed aggregated data by treatment arm during trial conduct, and all CDC staff was blinded to the regimen throughout implementation. Next slide, please. Primary safety outcome was proportion of participants with all-cause grade 3 or higher AEs during study treatment. It was comparable for Rifapentine, Moxifloxacin, and control regimens, around 19%, and difference was not statistically significant. And Rifapentine arm had significantly lower proportion of severe AEs at 14%. Next slide, please. So this slide shows time to first grade 3 or higher event during treatment or follow-up. Rifapentine-Moxifloxacin arm, shown in green line here, had slightly higher rate of grade 3 to 5 events compared to control arm, which is shown with blue line, but the difference was borderline not significant, as assessed in a log rank test. In opposite, the rate of severe events in rifapentine only arm, shown in red line, was non-significantly lower than in control arm. Next slide, please. So I just remind that primary safety outcome comparison was grade 3-5 AEs during study treatment, which was 17 weeks of data for experimental regimens and 26 weeks for a control regimen. These time points are noted with crosses on the graph. And you can see similar proportions of participants with an event by week 17 in Rifapentine-Moxifloxacin and week 26 for control arm. Next slide, please. This table provides some granularity on the types of grade 3-5 events shown by major system organ class term, or SOC, and these events occurred during study treatment or up to 14 days after the last study dose to account for late toxicities. There was no seen any safety signal or particular disbalances in frequency of types of events across the arms. On the top of this table, you can see that only 2 classes of events were seen with frequency at 3% or higher in any arm. The blood lymphatic system disorders, mostly represented by afebrile asymptomatic neutropenias, and hepatobiliary disorders, all were hepatitis. So these were the only, the most common events. Errors show events of particular interest as side effects of the fluoroquinolones. You can see that nervous system disorders, as well as musculoskeletal and connective tissue disorders, were quite well balanced across the arms and were very infrequent. One participant in Rifapentine-Moxifloxacin arm developed knee tendinitis and was discontinued from study treatment by PI's decision. There were 3 cardiac disorders, and all of these were in Rifapentine-Moxifloxacin arm. One participant with event that was related to study treatment had QTc prolongation to 460 milliseconds with palpitations and was discontinued from treatment after taking 3 doses of the study drugs. This was the only case of QTc prolongation in study 31 that we are aware of. 2 other cardiac AEs were congestive heart failure and right ventricular failure, reported to be unrelated to the study treatment. Next slide, please. In the secondary safety analysis, Rifapentine-Moxifloxacin arm had slightly higher proportion of treatment-related severe events compared to control arm. However, this could be related to open label trial design, as relatedness was determined by the site investigators who were unblinded to the study regimen. Proportion of participants with treatment discontinuations noted as tolerability here, at least 1 serious adverse event during study treatment, and all-cause deaths during study treatment were slightly lower in participant on both rifapentine arms compared to control arm. Next slide, please. Overall, 36 participants died during treatment or follow-up. All-cause mortality during treatment and follow-up was similar across the arms: 1.4% in control arm, 1.3% in rifapentine arm, and 1.5% in Rifapentine-Moxifloxacin arm. Control arm had slightly higher proportion of TB-related deaths, 1%, and Rifapentine and Rifapentine-Moxifloxacin had similar proportions, 0.5 and 0.4% respectively. Next slide, please. This slide shows deaths during study treatment by MedDRA preferred term. There was no detected safety signal in disbalance of any types of events. Control arm had more deaths during treatment, 7 versus 4 and 3 in the Rifapentine arms, respectively. Potentially, this was observed due to longer duration of standard treatment. One participant in Rifapentine-Moxifloxacin arm died of thrombotic thrombocytopenic purpura, which is known side effect of rifamycins. It was considered as a suspected unexpected serious adverse reaction or SUSAR event due to unexpected severity of event. Congestive cardiac failure in Rifapentine-Moxie arm was considered to be unrelated to the study treatment. Next slide, please. So this is a similar table, but shows deaths after completion of the study treatment. Again, there was no seen any particular safety signal, no disbalance in events led to fatal outcome to across the arms. Mortality was slightly high in Rifapentine arms as compared to control arm. But the numbers were very small. Mortality was slightly, and there were 2 unattended deaths seen. So you can see that in Rifapentine-Moxifloxacin arm there was 1 right ventricular failure death. It was considered to be unrelated to the study drugs. And also you can see it's 1 hepatitis, 1 liver death in Rifapentine-Moxifloxacin arm, which occurred in participant who was referred to National TB Control Program and restarted treatment there after permanent discontinuation of the study treatment. Next slide, please. So this graph summarizes survival time in 3 study arms. And time to death did not differ significantly across the arms. Next slide, please. So finally, we looked at liver chemistries across treatment arms in several ways, such as proportion of participants with any transaminase, ALT or AST value increased 3 times or more times upper limit normal during study treatment, shown in the top row. Proportion of participants with high transaminase thresholds of 5, 10, and 20 times respective increase over upper limit normal. And finally, combined transaminase and bilirubin elevations shown in the bottom row. So you can see that it's actually slightly high proportion of participants in control arm had transaminases increase. Not shown here, but we found that proportion of participants with grade 3 or high bilirubin was higher in Rifapentine arms. So control had 1% and Rifapentine and Rifapentine-Moxifloxacin arm had 2.4 and 3% respective grade 3 bilirubin increase. So we also examined these 25 participants who met Hy's law criteria shown in the bottom row here. And we found that these participants did not have very high transaminitis and bilirubin had been modestly increased. So this was not suggestive of severe hepatobiliary injury. So next slide, please. So in conclusions, in safety analysis, both high-dose Rifapentine regimens were safe and well tolerated. Next slide, please. Here are our acknowledgements again to funders, partners, and patients. Thank you.
Thank you, Ekaterina. And I know we're at time. I'm going to squeeze in one more question to Dr. Armitage, and then I'm going to do a quick wrap up and give an option for those of you who might have time to stay on a little bit longer. But Lisa Armitage, you're a TB consultant who advises other TB clinicians all the time. I'm curious to hear your opinion about whether you're worried about hepatotoxicity, particularly if for when it gets rolled out into real world settings rather than the clinical setting.
Yeah, honestly, I'm not. I feel like the studies, and they've been very transparent, with the findings. You can see that, comparing the Rifapentine-Moxifloxacin arm to the standard treatment arm, the outcomes are very similar, and I felt like they broke down on many levels that that give me a lot of comfort with it. I don't anticipate anything really different, and it's really refreshing to see that there has been a lot of kind of clarity and transparency in what the findings were. So I really don't anticipate any surprises.
Thank you so much. And I see we are at end of time for this session. I want folks to know that we will be recording this. I want to take a moment to thank all our speakers and discussants for sharing their time and expertise with us today. The impressive study really shows us it's possible to get to 4 months for treatment of TB disease, and perhaps even shorter. As we move forward, we'll continue to learn how to optimize outcomes. We are going to continue with some additional Q&A at this point for folks who are willing and would like to stay on the line. And so with that, let me start with this first question.
I know we're getting some questions about CDC national guidance and timeline for that, which I'm happy to address in a bit. Before we go there, I wanted to ask this question of Dr. Ashkin. What are some of the practical considerations from your perspective you think a TB program should keep in mind in sorting out whether or how to make this new regimen available?
I think there's a couple of things that happened in the study that I think we need to be prepared for. The first, I think, is important, is that we need to have rapid fluoroquinolone susceptibilities available. One of the things about this study is that every one of the patients who were included in the study had access to either rapid GYRA susceptibilities or regular susceptibilities. And as per the study, if they were resistant, they kind of threw them out of the study. But in reality, we can't just throw these patients out. So I personally believe, A, is that we really do need to have GYRA rapid testing. And that means the ability, if we do have it, to start Moxie and Rifapentine on day 1, which is what we have to do. As you know, in most states don't even do routine fluoroquinolone testing, but in those that do, at least let's say in Florida, every year we find about 3 to 5% of all of our patients, even though they're susceptible to INH and rifampin, they're resistant to the fluoroquinolone, which means these patients will not be accessible. The other things I think that we really need to worry about clinically is the cost. If you look at the cost of Rifapentine here, one of the big things that makes this study great is the high dose Rifapentine. But interestingly enough, using the high dose Rifapentine, even though we're only using it for 4 months, the average cost is about $1,200 for a 4-month regimen using the regimen used. And that's by 340B pricing, at least based on what we pay in Florida. Whereas Rifa costs about $400 for 26 weeks. So you're talking about a 3 times increase in cost. And that doesn't sound like a lot, but it is. I mean, if you look at Florida, where we have 500 cases, let's say a year, our cost then of drugs would go from somewhere around $200,000 to over $600,000. So that's significant. I think we need to look at it. I know you're going to say there's cost savings with DOT and stuff, but the bottom line comes down to most of us are using more and more video DOT, which is now becoming cheaper and less staff. So I think that's definitely going to be, need to be looked at. And then obviously the cost of contact investigations that may need to be done for treatment in the patient's fail. But I think the most important thing from my perspective, to be honest with you, is the loss of 3HP, potential loss. So one of the problems we have is that because of all the nitrosamine issues and concerns, Rifapentine is actually pretty hard to come by, or it's relatively hard to come by. And now we're using 9 times, almost 10 times the dosage of Rifapentine to get one person through. And in Florida, we're using a lot of 3HP with great success. So now, if we have a limited amount of Rifapentine, we're going to need to figure out who prioritizes, who gets that. And if we use all of our Rifapentine towards the 4-month Rifapentine trial, then we're potentially not going to have enough for 3HP. And I understand that we have rifampin for 4 months, but our 3HP is really, really being tolerated well with very, very high success rates. So for at least these are the things that my program here in Florida is going through and considering to try to figure out how we're going to approach this.
Thanks, Dave.
I do want to just give a chance to Dr. Dorman, if you can comment on some of what Dave said about cost.
Yeah, thanks so much, Neela. Dave's got some very good points about cost, and he is absolutely right that at the moment, Rifapentine is more expensive, and the supply chain is not robust at the moment. And these are important things that we all face. But I would also put forward that these are temporary things that are fixable, can be remediated. If we think back to when Rifampin first came out, a couple of decades ago, it was because of cost reasons reserved for certain patients and not used widely. And I think we've come to find out that that was not the best judgment and not in the best interests of patients and patients' health. And so I think, costs are things we all face, but costs are a temporary thing, and we together can push and drive the field and the market forward. So I don't see cost as a medium or long-term issue in this case. And I think the supply chain issues they are surmountable and they will be overcome.
Appreciate those comments. While I have you, there was a question about real-life monitoring. Do you think the regimen would require any unfamiliar methods for monitoring for the well-being of patients during and after treatment in a public health clinic?
I can try to take that one on as well. Beyond fluoroquinolone DST, one might wonder about EKGs or ECGs. The study did not incorporate those, and that was in alignment with FDA and other regulatory things. And by and large, we did not have safety signals, save for the one participant that Katya mentioned. So my own perspective is that EKGs would not be warranted. And then the other monitoring really was comprised of blood chemistries and blood counts that programs do on a pretty routine basis. I think that there would have to be some, settling in on how frequently to do those things. But I think that they are fairly routine studies.
Thanks so much. I'm seeing this question about how soon the regimen will be approved or adopted for clinical use. Will CDC publish treatment recommendations? I'll go ahead and tackle that one. There has been draft guidance that's been written and reviewed by an external expert panel, and this proposed guidance will be presented to the Advisory Committee on the Elimination of TB, or ACET, December 14th or 15th. This is a virtual meeting open to the public, and I'll post the link in the chat. But typically the call-in information and agenda are posted about 2 months before the meeting, and we anticipate that finalized guidance to be released early next year. I'm going to punt this next question to Payam. How should we think about intensive and continuous phase with this regimen? How many months would each be?
Thanks, Neela. I guess, traditionally the intensive phase has more drugs. That's why we call it intensive. The continuation, fewer drugs. So for the purposes of this 4-month regimen, the first 2 months are what we would call the intensive phase, and the second 2 months with 3 drugs instead of 4, the continuation phase. Operationally, I don't know that there's a big impact on that transition other than 1 less drug. We did adjust pyrazinamide dosing if there was a weight band change, and that's probably worth noting, which is what we do in routine practice anyway. If there was a weight change during treatment, the pyrazinamide dose was adjusted according to CDC guidelines.
Great, thanks. And I'm going to direct this question to Dick because of his close attention to all of the subgroups. But what are clinical characteristics or demographics for which you might not want to use this regimen, if any?
Yeah, well, I think what we found is that the subgroup analyses showed it was a robust regimen across the board. So there might be some subgroups for the Rifapentine regimen without Moxie that you might consider using that regimen for, but I don't want to get too far ahead on that. But I don't think there are subgroups where we can say, oh, this would not be a good choice for that subgroup. There was no interaction with any of the subgroups that we looked at. And in a couple of the subgroups, it looked like the regimen was, if anything, a little better, but we're not claiming that it was statistically better. So I don't think there are any subgroups to avoid. I do want to, if I could take a moment to comment on Dave Ashkin's point about the supply. I feel sorry for Dave because he's got a half-empty glass, and I have the solution because I have a half-full glass. So if we put them together, we'll have a full glass. The supply of Rifapentine is improving, weekly, and the nitrosamine issue has been dealt with. There was a delay there, and now the supply is becoming more is growing greatly, and there's now a generic manufacturer on the global market, not in the U.S. market, but that will make more product available for the U.S. And there's about to be there will be a second generic manufacturer on the global market within the year. And so the supply is growing greatly, and that's good news for all of us. So there should be enough to go around.
Thanks, Dick. And I'm just seeing a comment in the chat about what about excluded participants? I guess would those be folks who might be reserved about using the regimen? And just to go on with that, also questions about how many of the excluded participants ended up having fluoroquinol resistance, and what's a reasonable or possible treatment approach to those patients found to have fluoroquinol resistance, or what happened, at least in study protocol, as far as you know?
So there were very few people excluded for fluoroquinolone resistance. I don't know the number off the top of my head, but it was a very small number, and they were late exclusions. There were more exclusions for isoniazid resistance, which was not detected at enrollment because people were all diagnosed by gene expert for the most part. For people with fluoroquinolone resistance, the answer is control, the standard of care. It works just fine if there's fluoroquinolone resistance, assuming that there isn't any other resistance. One of the questions earlier in the chat or in the Q&A was about extrapulmonary disease, and as we mentioned, we excluded people with CNS or bone and joint TB. We didn't exclude other pulmonary TB as long as they had pulmonary TB, but only a handful of individuals had extrapulmonary TB that was confirmed. We don't know about unconfirmed, pulmonary TB, extrapulmonary TB. But with the exception of bone and joint and CNS TB, there's no reason to think that the Moxie Rifapentine regimen wouldn't work just fine based on extrapolation from what we do for extrapulmonary TB in general. So it should, that's a group that we excluded, but there's no reason to think that the regimen can't be used for most extrapulmonary TB.
No, thanks, and for you and all the experts on the line, we appreciate that you guys are speaking to some things that may be uncertain or that wasn't specifically addressed in trial, and I recognize that some of those answers can change over time. So with that in mind, I'm gonna see if I can ask Lisa Armitage about this one. There's a question about whether this would be a more liver-friendly TB treatment for patients with cirrhosis, perhaps because there's shorter exposure. Could the regimen even be used in patients with renal insufficiency? How comfortable would you feel with this regimen in those populations, Lisa?
I think I don't, in looking at the side effects that were noted in the trial, I don't think I would feel any more comfortable using kind of the standard 6-month regimen than I would the shorter course regimen. And there's, interestingly, you're pulling out ethambutol, which is cleared by the kidneys. Now you have just one, which is PZA, and we know how to dose that in patients with renal failure. I'm sure, I mean, it wasn't something that was studied, but would I feel comfortable doing it, giving a patient PZA 3 times a week instead of daily? Absolutely. I think that could be an easy substitution. Every patient I have with cirrhosis or liver disease, I'm watching like a hawk, you know, either way. I probably would be a little concerned about the PZA in patients who have liver disease, but I mean, really adding moxifloxacin in place of ethambutol, it doesn't do anything to the toxicity profile in my head, except just patients who may have cardiac disease.
Thanks. I'm moving to some technical questions that we got from audience, both on pre-registration and here. And this one sounds good for Payam. What was the time to conversion to a negative from a positive, either AFB smear or culture on this regimen and study?
I just actually put a response in the chat about that. Aside from time to culture conversion, the proportion that were culture negative at 8 weeks was higher in the Rifapentine Moxie arm as compared to the control arm, 78.5% in the Rifapentine Moxie versus 63% in the control arm. So there's a question about sort of airborne precaution and time to sort of culture negativity. That's one measure of this. At 12 weeks, we had about a 90.5% culture conversion with the Rifapentine Moxie arm versus an 86% conversion in control arm. But by and large, time to culture conversion on liquid media was very comparable, frankly, across all the arms. And you know, that's one of the learnings from the trial. From a trial methodology perspective, we use these culture conversion time points as surrogates for some of our decision making, and they actually aren't terribly predictive of what's going to happen. And it needs a bit more sophistication than just a culture conversion. But by and large, the new 4-month regimen seemed to convert a larger proportion of the population by 8 weeks than control arm.
Thanks, Payam. I'm going to direct this question to Katya. What was the maximum amount of time allowed for completion of treatment in the study?
So in terms of the study protocols, there was no pre-specified allowed time to complete the treatment. For statistical analysis per protocol, completion of treatment allowed 2 weeks during intensive phase, 2 additional weeks, and 3 weeks during continuation phase to meet criteria of per protocol analysis. So additional 5 weeks to complete full treatment regimen.
Thanks, and this might dovetail with this, and maybe I'll direct it at Lisa Chen. If there is a clinical assessment that treatment needs to extend beyond 4 months, so a patient maybe who has microbiological failure or extensive disease burden, what would you consider doing with the treatment regimen? Again, recognizing.
All right, no data zone. Yeah, right. But I think, you know, I would consider that this is no different than what we do in our daily, you know, care of TB cases, and that you put on your good clinician cap and you think about, you know, what are the things I'm following that will help me decide when to extend. And I think your idea about transition is, you know, I suppose, again, in a no data zone, I would revert to what would be control. And I would apply all the lessons I know about, you know, if someone appears to be failing, you know, you would continue, you retest their DST, you would, you know, extend treatment. If it was someone who is diabetic with a bad cavitary disease, you know, maybe, you know, I would revert to what we do have data for and probably use a standard treatment and out to 9 months. You know, there's so much of what we do has not had the good fortune of good data. I think that the starting point of having good data on what we can do to shorten is really exciting. And then we use our good common clinical sense for when things don't go the way we'd want them to do in the 4-month. And so nothing, even the failures that Dave so rightly pointed out are of concern, but you would be watching that patient and then you would be deciding, is this someone who I need to treat longer because they're responding in a slower way or not the way I wish they were. So I would revert to control and then add all the other normal clinical decision-making to get them to cure that we do with our routine cases.
Thanks, Lisa. I'm going to direct this question to Payam. We had a question about what are your concerns about rifapentine tolerance and specifically hypersensitivity reactions with this regimen, similar to concerns that were brought up when rifapentine was given in the 3-HP regimen.
Thanks very much, Neela. Yes, indeed. I think we all know that the rifamycins have been for a long while known to have these hypersensitivity reactions, all of them, rifampin not excluded. The experience with 3-HP is also notable, and we think there's an aspect of that is the fact that it's an intermittently dosed regimen. We didn't see signals of hypersensitivity that were were concerning in study 31. It's daily dose, it's higher dose, it's not intermittent. So maybe there's something about that there. We don't anticipate this regimen causing those kinds of hypersensitivity reactions that had been seen with 3-HP, but it's still very much part of the drug's profile of adverse events and has to be monitored.
No, and actually, while I have you, I know there's a question in the chat about why was it that the study investigators chose to look at rifapentine rather than the more widely available rifampin as the drug of choice in evaluating treatment shortening?
Yeah, sure, thanks. I answered this in the text as well, but there's a long journey for rifapentine, as you all know, from study 22 and actually even before then, some BMRC studies. It has been found to be more potent than rifampin insofar as an MIC. It's 1 to 2 dilutions more potent there. It has a longer half-life, possibly as long as 5 times longer. So that all these pharmacokinetic features of the drug mean that it corresponds to a greater potency, a longer duration of exposure to sort of kill off those bacteria that are in little niches and hiding. Where it can get to them. And then there was an extensive series of murine studies, preclinical studies, and this TBTC conducted really excellent dose ranging studies to build the case around rifapentine. So that's really what the underpinning of that choosing the rifapentine are. With that said, there's other research groups that are looking at higher dose rifampin, and those trials, one of them will, I think, report next year. And we can see how higher dose rifampin does in relation to, treatment shortening at 4 months as compared to this rifapentine arm. Obviously not directly comparable, but we can sort of look at the trials and draw some judgments from them.
Absolutely. No, thanks. This question, I'm going to turf over to Susan because you do have experience both in these South Carolina TB clinics as well as being closely involved in the study. There's a question about why did study investigators choose to evaluate resistance the way they did? How do investigators view the role of molecular susceptibility testing versus reliance on DST?
Yeah, good question. Thanks for that, Neela. Partly we, as a study group, we had to deal with the types of technologies that were available several years ago now when the study began. So we actually allowed either molecular or phenotypic drug susceptibility testing to be used at the sites. Or let's see, they all had to do phenotypic testing, but they could do molecular testing up front in order to sort of guide eligibility decisions. So we allowed for molecular use, but required growth-based phenotypic testing. And partly it was a response to what was available at the time. I think if we were to do the study now, the diagnostics landscape is a bit different, as you know, and so there might be different decisions that we could make.
Yeah, no, absolutely. The fact that it's come out and that we still have a landscape where it perhaps could even be better utilized or targeted is really excellent. Okay, I'm going to direct this question to Katya because you had talked about some of the safety profile. There's a question about if specifically pyrazinamide, if pyrazinamide wasn't tolerated by a trial participant, were other agents continued and then treatment extended to 9 months? You know, it's quite specific. I don't know how much pyrazinamide intolerance there was, but perhaps you could address.
Yeah, thank you. So it was very, very little intolerance of PZA, but if participant had it, no extension of other drugs were allowed. So any treatment extension would be considered an unfavorable outcome. So in this case, participants would stop the study drugs, transfer it to appropriate regimen, and continue follow-up in the trial.
Thank you so much. I'm going to direct this next question to Al. What do you think about converting a person who just started traditional RIPE over to this regimen? If people walk away and get really excited and say, you know what, I'm going to switch over my patient who's on a 6-month regimen to this newer regimen, what's kind of your gut reaction to that? Again, recognizing it's a data-free zone.
I know.
You might get
well, I think I would try to stick as close as possible to the study. And, you know, if I didn't start with this, I probably would just complete what I started and keep it clean for now until we know more about how the regimen performs. Now, going back to the earlier question you had about what to do if it's a slow responder, I see no reason why a clinician can't decide to extend this regimen. By the way, I think we need a nickname for it. You know, it's a continuation phase is 3 drugs. So if it's a slow converter or slow responder, I would have no problem keeping it going beyond the 4 months. So that's another approach I would perhaps advocate.
No, thanks, Al. A little bird is telling me that that Dick, you might know something about the Union conference and some additional presentation data there that might be of interest to this group for those folks that might be attending the International Union TB meeting.
Oh, I don't know what bird that was, but there is a there's a symposium of the study 31-5349 data at the Union. I'll get to the exact date and time in just a second, but it's going to include the 18-month follow-up results, as well as a lot of the PK. And it's going to be on Thursday, the 21st of October at 10:30 Eastern Time in the US, 4:30 European Time. And there'll be presentations on PK, PD, and the 18-month results. So it should be very, very fascinating.
Thank you so much. And I did put a promise in the chat to do a hard stop at 4 p.m. out of courtesy to our speakers and discussants, but just want to express huge gratitude for all of you, not only presenting all the data today, but being willing to answer questions from our frontline practitioners. And thank you to the audience for all your excellent questions. And stay tuned. We'll continue on this journey together. Thanks so much.
Thanks, Neela. Thanks, everyone.
Thanks, Neela.
Thank you.
Thank you.